So, good evening, everyone. Today we are going to talk about host pathogen interaction. Week 7, we are going to, revise. So, if you have any doubt, let me know. So, I'm Ananya Chatterjee. I'm a Prime Minister Research Fellow in IISc Bangalore, and I'm discussing about the course host pathogen interaction, where course instructor is Professor Himanshu Kumar, Professor of Department of Biological Science, IISR Bhopal. So the problem one, which of the following is not a member of RLR RIG1 like receptor family? Answer will be, so options are RIG1, MDA5, TLR9, LGP2 and none. So the answer is which of the following is not a member of RLR, or the full form is RIG1 like receptor. Option C, TLR9. Which of the following is not a downstream effector molecule activated by the STING pathway? IRF3, NF kappa beta, TVK1, CGMP, STAT3. Answer is STAT3. The CAR domain in RLRs is essential for oligomerization. Pro-interleukin 1 beta and pro-interleukin 18 are inactive forms of cytokines that are clipped into their active forms by caspase 1. Inhaled P3 can sense RNA, DNA, cytosolic toxins, silica, all are correct. Option is all are correct. SLR14 that acts as powerful immune adjuvant and induces anti-tumor responses is an antagonist of RIG1, MDA5, M2, LGP2, TLR7. SLR14 that acts as a powerful immune adjuvant and induces anti-tumor response is an antagonist of RIG1. Peptidoglycan from gram-positive bacteria can act as ligand for option 1 TLR1 and TLR2, RIG1 and MDA5, NOD1 and NOD2, NAPE and NLRC4, M2 and LGP2, NOD1 and NOD2. BIR domain is an exclusive characteristic of CIITA, NAIP, NLRP2, NLRC4 and NLRP5. So BIR domain is an exclusive characteristic of NAIP. Which among the following statements related to RLR are correct? Alternatives in RNA metabolism cannot generate RLR stimulatory RNAs. Mutations in RLRs causes spontaneous signaling in the absence of virus infection. Mislocalization of mitochondrial RNA into the cytosol leads to MDA5 activation. Option 1, 1 only, 1 and 2, 1 and 3, 2 only, 1, 2, 3, 3 only, 2 and 3, none of the statements. Answer is 2 and 3. Which order are not associated with mutation in NLRP3? Options: Markel-Wells syndrome, Aicardi-Gautier syndrome, familial cold autoinflammatory syndrome. So answer is option 2 only. Properties of immunogen contribute to immunogenicity. Foreignness, molecular size, chemical composition and complexity, ability to process and present antigen or immunogen on MSC. Foreignness, immunity of the host do not develop immune response to self-antigens. Degree of the immunogenicity depends on the degree of its foreignness. For example, bovine serum albumin is not immunogenic when injected into cow, or less immunogenic in goat, but is when injected into chicken it is immunogenic. Some macromolecules are highly conserved throughout the evolution and display little immunogenicity. Example: cytochrome C, collagen. Some exception, corneal tissue and sperm, they will be immunogenic. Molecular size, active immunogens greater than 100,000 Daltons, poor immunogens less than 5,000 to 10,000 Daltons. So, chemical composition and complexity. 4 levels of protein organization structure. Primary structure, amino acid sequence of polypeptide chain. Secondary structure alpha helix and beta plated sheets. Tertiary structure there are domains monomeric polypeptide molecule. Quaternary structure dimeric protein molecule. Chemical composition and complexity. Polymers composed of multiple copies of amino acid and sugar tend to be more immunogens. Lipids are haptens and need to be conjugated with carrier to produce antibodies. Important for assays for detection of some steroids and vitamins. Susceptibility to antigen processing. So bacteria is entering and it's getting phagocytosis. So it will be inside lysosome, and then tumor or viral antigens, it will get processed in vesicular pathway, and then it will be showcased by MHC. This is MHC class 1, this is MHC class 2 pathway. Large insoluble macromolecules are more likely to be phagocytosed for processing D amino acid or L amino acid polymer, as L amino acid is dominant in living system and processed by host enzyme. It is immunogenic. Role of biological system in immunogenicity. Host genetic makeup, route and amount of challenging antigen, use of agents to enhance immunogenicity. Host genetic makeup of recipient host. Genes of MSC controls response to antigen. Genes involved in coding for antibodies. B cell and T cell receptor genes. Route and amount in which material is presented. Too low or high doses can induce tolerance. Single dose is often not enough. Booster is needed. Route, different routes. Intravenous, intradermal, subcutaneous, intramuscular, intraperitoneal, intranasal. Use of agents, adjuvants to enhance immunogenicity. Immunogenicity enhancing agent is known as adjuvant. Mechanisms not precisely clear, but some might activate PRRs. Water in oil adjuvants, fluids in complete adjuvant, antigen in aqueous solution, mineral oil, and emulsifying agent. Antigen is then released very slowly from injection site. Based on fluids, complete adjuvant also contain heat-killed mycobacteria. Use of agents to enhance immunogenicity. Fluids incomplete adjuvant, fluids complete adjuvant. So this prolongs antigen persistence. Aluminum potassium sulfate alum prolongs antigen persistence. The first two enhances co-stimulatory signal. It induces granuloma formation, but it doesn't stimulate lymphocyte non-specifically. Epitopes. Antigen determinants recognized by B cells and T cells. B cell epitopes tend to be on the outside of the antigen, for example, the hydrophilic amino acid on a protein surface. T cell epitopes from proteins derived from enzymatic digestion of peptides and then association with MHC. Glucagon, a hormone when used to immunize The hydrophilic amino acids on a protein surface. T cell epitopes from proteins derived from enzymatic digestion of peptides, and then association with MSC. Glucagon, a hormone, when used to immunize, the B cell and T cell epitope differ. B cell epitopes have characteristic properties. Located on surface of immunogen, accessible to antibody. For proteins immunogens, the epitopes can be sequential or non-sequential, referring to amino acid sequence depending on the protein folding. Sequential epitopes recognized by B cell repertoire. B cell repertoire, B cell, original antigen, schematic primary structure. This is a different, like sequential epitopes: clone 1, clone 12, clone 12, clone 10, clone 9 with different genes, and so on. Non-sequential epitopes: B cell receptor, B cell, original antigen, recognition of proteins and peptides through discontinuous epitopes on them. These are discontinuous epitope. See, with yellow and pink over here, with this orange color and magenta color over there. This is discontinuous. How in each case two widely separated portions of the protein come together in space in their three-dimensional conformations and get recognized as single epitope. Okay. Comparison of antigen recognition by T cells and B cells. Comparison of antigen recognition by T cells and B cells. Characteristics: interaction with antigen. B cells involves binary complex of membrane Ig and antigen. T cells involves ternary complex of T cell receptor, antigen and MSC molecule. Binding of soluble antigen, B cells yes, T cells no. Involvement of MSC molecules, B cell none required. T cell required to display processed antigen. Chemical nature of antigens: protein, polysaccharide and lipid. T cells mostly proteins but some lipids and glycolipids presented on MSC like molecules. Epitope properties: accessible hydrophilic mobile peptides containing sequential or non-sequential amino acids. T cells: internal linear peptides produced by processing of antigens bound to MSC molecules. Production of monoclonal antibody. Hybridoma technology. Step 1: immunization of mice and selection of mouse donor for generation of hybridoma cells. Antigen: intact cell or whole cell membrane or microorganism plus adjuvant. So these are antigen. Antigen is getting injected. Antibody titer reached in serum and spleen removed, where you will get the source of cells. Screening of mice for antibody production. After several weeks of immunization after injecting antigen, you are selecting serum antibody titer. You determine serum antibody titer by ELISA flow cytometry. Option 1 is titer 2 slow, option 2 is titer high. So titer 2 low will boost and titer high boost pure antigen. So after like in both the cases, you will boost it with pure antigen. Preparation of myeloma cells and with AGPRT minus, high viability and rapid growth. Molecular basis of hybridoma technique or hybridoma selection. Dihydrofolate will get converted into tetrahydrofolate. Precursors: de novo synthesis nucleotides to DNA. And salvage pathway, what happened? Hypoxanthine gets converted to nucleotides, and then with the help of AGPRT, the path is first. So pathways for the synthesis of nucleotides: AGPRT, hypoxanthine guanine phosphoribosyl transferase. RTK is thymidine kinase. Step 4: fusion of myeloma cells with immune spleen cells and selection of hybridoma cells. So spleen cells, peg fusion, myeloma with myeloma cells, hybridoma cells. So spleens gets fixed with fused with myeloma cells. Plating of cells in HAD selective media. Scanning of viable hybridomas. Cloning of hybridoma cell lines by limiting dilution or expansion. Option A: clone each positive culture. Option B: test each supernatant for antibodies. explained positive clones. Application of monoclonal and polyclonal antibody. Monoclonal antibody basic research, immunoblood, immunohistochemistry, cell analysis by flow cytometry, diagnosis, pathogen detection, malignancy test, diagnosis of array of infection and autoimmune disease, therapeutic use, cancer treatment, autoimmune disease, allergy treatment, polyclonal antibody basic research, immunoprecipitation, chromatin immunoprecipitation, immunoelectrophoresis, radial immunodiffusions, diagnosis, histopathological analysis, therapeutic use, passive immunization, antivenom. Beginning of conceptualization of soluble component in host defense. Emil von Behring talked about serum antitoxins. This is Paul Ehrlich and his drawings of the formation of effector functions of antibodies according to the side chain theory. So, how do discovery of antibodies happen? Known since late 19th century that antibodies are in serum in fluid phase that remains after blood clotting. Antibodies are also found in other secretions. Serum protein electrophoresis, Arne Tesselis and Elvin Corbett experiment 1939. Absorbance and migration distance. So here you can see these proteins are getting separated on the basis of absorption and migration distance. This is albumin. These are alpha, beta, and gamma globulin. So in gel you can run the gel and see. Molecular compositions. Antibodies are heterodimers. Okay, heterodimers. So two light chains around 22 kDa each and two heavy chain 55 kDa each. First 110 amino acid of amino terminal end of heavy and light chain vary depending on antibody specificity. Antibody structure and function. So here you can see these two are heavy chains and these two are light chains. Effector function: opsonization, complement activation, antibody dependent cell mediated cytotoxicity or ADCC for killing of cells. Soluble antigen bind with antibody and form immune complex. Clues for basic antibody structure. So this is heavy chain and light chain. So disulfide bonds are there in between two heavy chains and light chains. So you see, if you do pepsin digestion, you will have this FC fragments, okay, along with this disulfide bonds of 100 kDa. This is called FAB2. If you do papain digestion, you will have Fab fragments and FC fragments. And marcaproethanol reduction will give you H chains and L chains separately. ठीक है. These are the gel after you SGS page gel after you run. Immunization of goat with antibodies from rabbits. Immunization with Fab fragment. Interaction with both heavy and light chains. Immunization with FC fragment interaction only with heavy chains. Sequence of antibody required a purified source of antibodies. B cells from multiple myeloma patients. Different digestion procedures reveal different fragments. Fab2 still shows antigen binding capability. Okay. We will learn about light chains and heavy chains more. Before that, if you have any doubt, let me know. given as GNBPs. Which of the following is microbial sensors in Drosophila? Okay, I will get back to you with this question later. I have to check one thing. So, which one is correct? Which one is incorrect? PRR PAM interaction can initiate inflammatory reaction, activate innate immune response, induces type 1 and type 3 interference, initiate a signaling cascade, initiate antibody production. So yeah, PRR and PAM. So this is pathogen recognition receptor and this is pathogen associated molecular pattern. This interaction alone directly can't initiate antibody production. There will be other cascade mechanism, right? There would be different kinds of cascade mechanism, and then induction of B cell, differentiation of B cell. After that only antibody production will be done. So the incorrect answer is this. PAMs are unique to each pathogen, found only in adaptive immunity. No. Responsible for creating antibodies. No. Conserved molecular patterns. Yeah. Conserved molecular patterns on pathogens. Cole's toxin, which was injected in cancer patient, was made up of tumor killing viruses, bacteria and bacterial products, bacteriophages, TNF alpha, complement proteins. It's made up of bacterial and bacterial products. Which among the following statements is correct? Microbes are 10 times more than the number of human cells. Microbiome present in the human body does not include fungi. Every day, an adult human swallow and digest about 1 liter of mucus. So, which among the following statements is not correct? Okay. So, 1 only, 1 and 2, 1 and 3, 2 only. So, option will be 2 only. So, microbes are 10 times more than the number of human cells. This is true. And every day, an adult human swallow and digest about 1 liter of mucus. This is also true. Which of the following statements regarding innate immunity is wrong? Innate immunity evolved 450 million years ago. Since the origin of the sponges, adaptive immune system evolved 700 million years since the first jawed vertebrates. Innate immune genes evolve slowly while pathogens evolve rapidly. Which of the following statements regarding innate immunity is or are wrong? Innate immunity evolved 450 million years ago since the origin of sponges, which is wrong. Adaptive immune system evolved 700 million years ago since first jawed vertebrate. This is also wrong. Innate immune genes evolve slowly while pathogens evolve rapidly. This is true. So option will be 1 and 2. Sorry. 1 and 2. Which of the following statements is or are correct? Lysozyme destroys the cell wall of bacteria. This is true. Dermis gives skin strength and pliability to resist abrasions that could introduce microorganisms. This is also correct. Sebum increases the pH of the skin to a level inhibitor to many bacteria. No, this is not true. Option will be 1 and 2. So today we will start complement system. we'll start complement system. So the term complement refers to a system of factors that occur in normal serum and is activated characteristically by antigen antibody interaction. So you need to understand that the term complement refers to a system of factors. So it's not a single component, but it's a system. That's why it's called complement system of several factors, okay, that occur in normal serum, and this system gets activated characteristically. So there is some specific mechanisms by which it gets activated by specially antigen antibody interaction. So if you discuss about history, scientist Buchner, German bacteriologist, was the first to observe that the bactericidal effect of serum was destroyed by heating at 55 degrees Celsius for 1 hour. Okay, so he observed that the serum do have bactericidal or killing bacteria activity, but it can be destroyed by heating at 55 degrees Celsius for 1 hour. That means you are destroying some factors which is present in serum. Scientist Pfeiffer, German bacteriologist, discovered the phenomenon of bacteriolysis. Observed that live cholera bacteria could be injected without ill effects into guinea pigs previously immunized against cholera. Okay, so he has observed that live cholera bacteria could be injected without ill effects into guinea pigs which were previously immunized against cholera. So there is something, some factor present inside those animals who were previously immunized against one specific bacteria, and if we again reinfect those, then what is happening? I'm sorry, I just forgot share my screen. Sorry for the inconvenience. Yes. So I was just discussing week 6 assignments. If you have any doubt in understanding week 6 assignment, you can let me know. I will just go through this question number 4 once again after some time, and rest of them I have discussed. So we have today we have started with complement system. So the term complement system, refers to the system of factors that occur in serum. So there is one multiple factors present in the serum that is activated characteristically by antigen antibody interaction. Okay, this kind of system is called complement system, and scientist Buchner, who is a German bacteriologist, was the first one to observe that the bactericidal effect of serum was destroyed by heating at 55 degrees Celsius for 1 hour. So, if you heat serum for around 55 degrees Celsius for 1 hour, then and if you treat bacteria with serum, so if you will be able to see that before heating there is a bactericidal effect, but after heating there is no bactericidal effect. So something is getting destroyed while heating. So scientist Pfeiffer, German bacteriologist, discovered that this phenomenon and discovered one phenomenon and termed this as bacteriolysis. So he observed that if you inject or if you infect one animal who was previously immunized against any virus, be it cholera, be it any not virus, bacteria, be it any bacteria. So he observed this in the case of bacteria. It can be valid if you have. It can be valid for virus if you have good immune antibody generated against it. Yeah, bactericidal effects means so there will be some factors which will help in killing bacteria. Bactericidal means killing bacteria, or it's like bacteria will get burst. Okay, So we can, I'll go. Yes, so what happens? He could see that after injecting live cholera bacteria in some guinea pigs who were previously immunized against that disease was didn't get infected. Okay, and plasma from these animals added to live cholera bacteria caused them to become motionless and to lyse. Yes, so this phenomenon is called bactericidal, as I was talking. So means, so their plasma has some factors, okay, has some proteins which you are going to add in live cholera bacteria, and then those factors will induce those bacteria to get lysed or burst or lifeless. Okay, this could be inhibited by previously hitting the blood plasma, but this bactericidal property you can inhibit if you hit this plasma before adding. That means you are denaturing the proteins or the factors which are important for bactericidal or killing like bactericidal means to kill bacteria. This activity is needed. So if you are destroying those, then you won't be able to see this bactericidal effect. So scientist Pfeiffer called this as bacteriolysis, and it became known as a Pfeiffer's phenomenon. So this is important to know what is Pfeiffer's phenomenon. Bourdon established that immune bacteriolysis and hemolysis require two factors. One is a heat stable antibody, and another factor is a heat labile factor alexin. Paul Ehrlich coined the term complement, replacing the term alexin, because this factor complemented the action of antibody. So here comes the story of complement system. Complement means complementary. Okay, so it's get activated in the presence of antigen antibody complex, and then it also first in the process. So there are two main thing is required to have bacteriolysis and hemolysis property. One is heat stable antibody, another one is heat labile factor alexin. This is further in future it was termed as complement or complement system. It is named complement system because it was first identified as a heat labile component of serum that complemented antibodies in the killing of bacteria. Consists of serum and cell surface proteins involved in defense against pathogens and tissue damage mediated by antibodies. Consists of serum and cell surface proteins involved in defense system involves in defense against pathogen and tissue damage mediated by antibodies. The complement system is a major effector of humoral branch of immune system. Plays major role in both innate and adaptive immunity. So what is the general properties of this complement system? These are non-specific. This consists of 5% normal serum protein destroyed in 30 minutes at 56 degrees Celsius, that is heat labile. Bind only to antibodies that have combined with their antigens. Only immunoglobulin M, immunoglobulin G, 312 fixed complement. Binds to FC portion of immunoglobulin. 30 proteins, C components per plus properdine plus control proteins. 9 different fractions C1 to C9. Complement is a complex of 9 different fractions C1 to C9. Fraction C1 occurs as calcium plus 2 ion dependent complex, which on chelation with EDTA yields 3 protein subunits C1, Q, R and S. Fractions are named C1 to N in the sequence of cascading reaction, except C4 comes before C2. So when a fraction acquires in enzymatic or biological activity, it is indicated by a bar over the component number. That is, if C1 gets activated, then C1 bar. They are mainly synthesized by hepatocytes, also produced by blood monocytes, tissue macrophages, and epithelial cells of the gastrointestinal and gastrourinary tract. Complement proteins made as zymogens, activation by cleavage. Example C4. So here you can see that from C4A, which is a smaller fragment, gets so sorry, yeah, so activated by cleavage and forms C4B, which is larger fragment. Exception C2, C2A is larger fragment and C2B is smaller fragment. Complement activation. So normally, yes, tell me. Yes, yes. So zymogens means, you know, those are enzymes, but it needs some cleavage activity. So this is a protein, okay, and so this is a zymozyme, but it's not active now. It's inactivated. So it needs to uh, break some bonds and then form two different fragments, and then it becomes like active enzymes. So here C4 is zymozymes, and then some cleavage is happening and it's forming C4A and C4B. So and it's becoming active enzyme, like which will do the function further in cascade mechanism. Yes, yes. So yes. So like cleavage is needed for their activation. I can show you some more example. Just a second. Mm. Yes, so if you see in Google, I will share the screen. Mm, Wait, my internet is poor today. Yeah, so if you just Google zymogens, you can find one image. Internet is super slow today. Anyways, yeah. So you can see this is a this is a protein, okay, or you can say enzyme, which is not active. So there are different environmental factors which are coming in the play in the action, and then either it is getting clipped or some autocatalysis activation is happening, okay, maybe like, so this is a protein, so some part of the protein is activated, somehow some bond formation or somehow something is happening inside. This is called autocatalysis. And if not autocatalysis, it can also uh, gets clipped into two substance, okay. So here you can see, so breaking a bond also can be called can be called, this autocatalysis activation. So here you can see this was zymogen, but after breaking a part of it apart, it is becoming active enzyme. So this is it. So complement activation, not, yes, yes, my internet is very bad today. I'm not at. I'm connecting with my cellular network. I'm not connected with Wi-Fi. I am sure. So I am recording the video where you will find the voice very clear, because I'm screen recording my screen and audio as well. So I'm uploading in my YouTube channel, which is already the link is shared with you all. NPTEL lectures by Ananya. You can, if you want to just revisit, you can. I am writing it in the chat section. So I will start again. Complement activation, normally present in inactive form in body. So this complement systems or the proteins associated with complement system are normally inactive. Activity is induced by antigen antibody combination. Components react in a specific sequence as a cascade. Cascade is a series of reactions in which the preceding components acts as enzymes for the succeeding components. Components are clipped in dissimilar fragments. Larger fragments join the cascade and the smaller fragments released in the medium possess some biological functions. Complement activation, lysis of cells and bacteria, promotes virus neutralization, opsonization, immune clearance, amplification of inflammatory process by increasing vascular permeability, release of histamine from mast cells. So one example, number 1, lyse bacteria by forming MAC. So complement system can lyse bacteria. Okay. Function 2, tag pathogens to enhance recognition. So some proteins associated with complement system can get attached with some specific bacteria to tag those bacteria and to enhance the recognition of those bacteria by phagocytic cells and distraction by phagocytes, which is called opsonization. Function 3, activate inflammatory response by triggering release of histamine from mast cells. So activation of inflammatory response, it can activate inflammatory response, so it triggers release of histamine. So basically complement systems interacts with complement receptors, and then some cascade mechanisms, it also interacts receptors present in mast cells, and then some different signaling pathways, some cascade receptors, cascade mechanism, it releases histamines or inflammatory molecules from mast cells. Function 4, enhance clearance of antigen antibody complexes. So suppose there are antigens which are getting presented by antigen presenting cells, and then antibodies are attached with it. Okay, so complement system comes into the action, and then it increases the clearance process. Basically it fasters the process of antigen antibody complex clearance. Complement activation pathways, so there are different pathways. We are just describing what it can do, but there are different pathways or signaling process, you can say that. Because why signaling? Because it's a cascade mechanism. One protein it's activating another protein, that protein is activating another substance, like that. So that's why it's called a cascade pathway. Or so we'll discuss complement activation pathways. C cascade is triggered off by 3 parallel but independent pathways. So complement pathways, also called C cascade pathway, differ only in initial steps. Once C3 activation occurs, the subsequent steps are common. 3 major pathways of complement activation. Classical pathway, activated by antibody bound to antigen. Alternative pathway, activated on microbial cell surfaces in the absence of antibody. Lectin pathway, activated by plasma lectin that binds to mannose residues on microbes. Central event in complement activation is proteolysis of C3 to generate biologically active products and subsequent covalent attachment of C3b to microbial cell surfaces or to antibody bound to antigen. So these are three basic pathway: classical, alternative, and lective. We have already discussed Pfeiffer phenomenon, and the complement system we have discussed about Bordet principle, and then lytic activity, bactericidal activity. Paul Ehrlich also conducted the similar experiment, coined the term complement. So complement term was coined by Paul Ehrlich. Components of complement, if you see, so this is the soluble proteins or glycoproteins. Glycoproteins means glycosylations happens in those proteins, so glycosylated residues are there, or glycans are present in the protein, synthesized mainly by liver hepatocytes and other cell types. So these proteins are synthesized mainly by liver cells, or also called hepatocytes, or other cell types. And we have discussed that 5% of serum globulins are complement proteins, and we also discussed that it circulates as inactive proenzyme, which is also called zymogens. So proteolytic cleavage happens, and it removes one inhibitory fragment and exposes its active site. So it can do lysis, we have talked about this. It can do opsonization and also activation of inflammatory response. Nomenclature of complement designated by, so complement nomenclature we also, yes, tell me. Opsonization is, so you have heard about phagocytosis. So opsonization is, yes. So opsonization means just, the enhancing the phagocytosis process further. So complement systems proteins are tagging those bacteria, and then bacteria in further is getting phagocytosed. Okay, so this is the same, yeah, the same part, but it's some other proteins are associated in helping the process. Can show you some picture of it if my internet works. So it's not like mandatory that only opsonization happens if complement system proteins helps, but some other factors also can get associated just to foster the thing. So if we see, opsonization can be because of different other factors. I will talk about it, but complement protein mediated opsonization, it's that you have to go in details that there are different factors, and by cascade. Oh, so okay, we'll talk about it later, because I have not introduced to you yet, like C3b, C4b, these things are not introduced yet. But if you see opsonization in overview, so there are several research paper you can go through, which are very much interesting. So pathogen opsonization, there is a video in YouTube you can see by complements how it. I'll just show you if it plays. It will be interesting. See if it's visible. So see, Phagocytosis of pathogen opsonized by complement system. So basically, one phagocytic cell, can phagocytose different pathogens. But if those pathogens are previously recognized by complement system, and those complement system help in recognizing the pathogen further to those phagocytous cells, so this process gets enhanced. So here it's happening that phagocytosis of pathogen opsonized by complement. So opsonized means it's getting engulfed by complement system. So this is one example of mannose binding lectin pathway. This is alternative pathway. Here it would be classic pathway. See, antibodies are, so pathogens are getting recognized by antibody, okay, and then it gets engulfed. These are different antimicrobial peptides. It's just breaking the pathogen. So yeah. So this is called opsonization. Nomenclature. Oh, something gets activation of innate cells. You can see this video if the video is running. So it's showing that it's showing that activation of innate cells against virus infect. Oh, sorry. So another video got played at its own. You can see the video. Activation of innate cells against virus infected cells. So this is an infected cell, and this is natural killer cell or NK cells. Yeah, it's nice, quick videos. Yeah, thanks. So complements are classified into 7 functional categories. Functional protein classes in the complement system: binding to antigen antibody complexes and pathogen surfaces. Example, C1q. Binding to carbohydrates structures such as mannose or N-acetyl glucosamine on microbial surfaces, which are mannose binding lectins, phycolins, properdin. Properdin is also called factor P. Activating enzymes C1R, C1S, C2A, and so on. Surface binding proteins and opsonins, example C4b, C3b. Yeah. So examples of, if you are asked example of some complement proteins which help in opsonization, your answer will be C4b, C3b. Peptide mediators of inflammation C5a, C3a, C4a. Membrane attack proteins or MAP, C5b, C6, C7, C8, C9. Complement receptor CR1, CR2, CR3, CR4. Complement regulatory proteins, there are a lot of names: C1 INH, DAF, CD59, and etc. So one example of alternative pathway, C3 protein undergoes spontaneous hydrolysis to C3H2O to initiate eventual deposition of C3 convertase on microbial surfaces. Classic pathway. C1q interacts with pathogen surface or with antibodies bound to the antigen surface. Lectin pathway. Mannose binding lectins and phycolins recognize and bind carbohydrates on pathogen surface. So there are different mannose binding lectins. It can recognize mannoses or glucose. You know, there are different viruses, different pathogens which has glycosylated protein. So they in their cases, glucose or mannose residues are exposed. So those residues they target, like those complement system target, like those are lectin proteins. So they have affinity towards glucose or mannose. So they bind to it and then recognize those antigens. So three pathways: lectin pathway, classical pathway, alternative pathway. In all this you can see that the cascade mechanism is activating different other complement proteins, example C4 C2, here also C4 C2 C3 in alternate pathway, and all pathways generate a C3 convertase which cleaves C3, leaving C3b bound to the microbial surface and releasing C3a. So what is happening? C3 convertase cleaves C3 and produces C3b and C3a. C3b remains bound to the microbial surface, but C3a, the other fragment, gets released. And then C3a and C5a recruit phagocytic cells to the site of infection and promote inflammation. This can be done. Then phagocytes with receptors for C3b engulf. So where you can see C3b gets remains attached with a microbe. So then phagocytes with receptors for C3b, there are cells which has receptor for C3b. They engulf those pathogens associated with C3b and then they degrade or destroy the pathogen. Completion of the complement cascade leads to formation of membrane attack complex or MAC, which disrupts cell membrane and causes cell lysis. This is the final event. So this is classical pathway. So here you will be able to see that different complement proteins are getting activated in cascade mechanism. So this is classical pathway, which is helped by antigen antibody complex. This is lectin pathway, involved mannose binding lectins, and this is alternative pathway. Ultimately they are all forming membrane attack complex. Complement classical pathway, antibody dependent, activated by antigen antibody complex, most commonly immunoglobulin M and immunoglobulin G. Early stages involve C1, C2, C3 and C4. C1 of classical pathway. C1 macromolecular complex C1QR2S2, two molecules of C1R and C1S and one molecule of C1Q. This is the structure of the protein or macromolecular complex. They have a stalk region and has head region. So here C1R and C1S, this two molecules, this is C1Q molecule. The total in the whole macromolecule is called C1 macromolecule. So this is the antibody structure and function. It's a Y-shaped. Here you have heavy chain, you have light chain. Heavy chain and light chain are getting attached by disulfide bond, and then two heavy chain as well is getting bonded by disulfide bond. So it has antigen binding domain and one FC region or constant region. So this has effector activity or different biological activity. So there are different antibody types: IgA, IgG, IgT, IgE, IgM. IgM is pentameric. IgA can be dimerized by J chain. This is a J chain. And this is IgG, IgD, and this is IgE. Yes, so this dimerization is required. So yeah, obviously if there is J chain, there is required. So sometime what happens if the antibody is dimer, okay, and J chain is making the dimeric bond. It's just in interaction of two monomers is happening. So if there is a macromolecular complex at it interacts, IgA is interacting, so J chain is giving a flexibility so that this is one IgA, this is another IgA is binding like this. So it's a big macromolecular complex and it's trying to find those epitopes in a very flexible manner. Okay, so yes, so this gives, yes, yes, yes. And in IgM, those are pentavalent. Pentavalent means if, like, you can see this five epitopes, like five binding motifs you can find, and there also one J chain is present, and those five pentamers are linked by disulfide bonds. So this is complement activation. Mm, okay, I will just see. So there is a science static paper. It's showing that the J chains are not necessary for formation of IgA dimers. However, they're essential for binding polymeric IgA to secretory component and are required for the efficient transportation of serum IgA into bile. So it gives solubility as well. So what is the role of J chain in secretory immunoglobulin formation? So the joining chain, which is joining two monomers, as is a small peptide expressed by mucosal and granular plasma cells, which regulates polymer formation, as I was telling, forming dimerization. But there is also role in making it secretory or solubility. So, how many Jechens are there? There are, although it's generally believed that pentameric IgM and dimeric IgA contain one Jechen per polymeric molecule. Some research group have reported that two Jechens for IgA and three or four Jechens for IgM. So it's conventionally one for each, but in some research paper you can find that they have reported two J chains for IgA. What is secretory? So if you are asked that what is the secretory component for IgA, secretory component is a proteolytic cleavage product of the polymeric immunoglobulin receptor which remains associated with dimeric IgA in seromucous secretions. Polymeric IgA binds to the polymeric immunoglobulin receptors on the basolateral surface. Okay, there's nothing is said here. Mm, there are different papers. Okay, okay, yeah. So you can ask questions, so that will give me more interest. Yeah, thank you for asking. So there are different papers you can read, and if you have any questions, we can discuss more. So there are one paper, structural requirements for incorporation of J chain into human IgM and IgA. So this is complement system, the cascade pathway in diagrammatic view. So what is happening here? C1q binds with antigen bound antibody. Okay. Then the C1q is the whole system, and then what happens? C1r activates autocatalytically and activates the second C1r. Both activate C1s. Okay. C1s cleaves C4 and C2, C4a and C4b, and in C2, C2b and C2a, C4 binds the surface near C1 and C2 binds C4, forming C3 convertase. Okay. This is C3 convertase, which is also called C4b C2a. C4b C2a, or in short C4b 2a. So this C3 convertase hydrolyzes many C3 molecules. Some combine with C3 convertase to form C5 convertase. So this C3 convertase can hydrolyze C3 into C3b and C3a, and also it can form C5 convertase, which is C4b 2a and 3b. The C3b component of C5 convertase binds C5, permitting C4b2a to cleave C5. Okay, so this is C5 convertase, which was C4b2a and 3b, and they binds to C5, which is basically C5b plus C5a, and then it's like which is cleaving C5 into this fragment. C5b binds to C6, initiating the formation of membrane attack complex. So this is C5b, from here C5b, C6, C7, and also it gets associated with C8 all together, like C5, C6, C7, C8 all together binds to C9 in the present in the membrane and forming membrane attack complex. So this is one. Sorry? Yes. So the whole thing you don't have to memorize, but if you read again and again, so there is one basic flowchart. You don't need to remember this C1q R2 S2. It's complicated. I have also forgotten. So you just have to remember that what is like. So there are some important steps. That is, you need to remember what is cleaving C3. So C4b C2a. You like once you read one, like so you need to see some videos, like diagnostic videos from where these pictures has been taken. You will get so once you understand, it will be uptaken by your brain. So what happens that after C1, it's going to, like, it cleaves C4 and C2. This much is fine, but you don't need to remember that C1 activates C1r and C1s. This is not required. This much specific question you won't get asked, and also it's not relatable. Where once you get question, you can just read some paper and then have a overview. So and then this is important. So what is C3 convertase? You can remember that it's made up of C4b and C2a. So C is not mentioned twice, just but it is C4b plus C2a. So like this, some important steps like C5 convertase, and then after that, after C5, you can see C5 is getting associated with C6, C7, C8 all together, and then C9 is present in the membrane already. It's tagging the antigen, so all together is coming here and then forming a membrane attack complex. So this is the story. So Submembrane attack complex you can see under electron microscope. So it basically forms a pore in cell membrane where ions and small molecule can freely pass through the pores, and if pore is getting generated in the cell won't be able to maintain osmotic stability. This is how it kills the target. So there are alternative complement pathway which is antibody independent. Component of innate immune system, early stages involve C3, factor B, factor D, properdin, initiated by cell surface constituent foreign to host, for example gram and gram positive bacteria. So initiators of the alternative pathway of complement system, you no need to recall all those 10 names. So there are pathogens and particles to microbial origin. There are different pathogens, different gram negative and gram positive bacteria. Non-pathogens, that is human IgE, IgG in complexes. So these are initiators of alternative pathway. In alternative pathway also you can see C3. Basically the complement system, you have to remember that the protein gets, these are in zymogens form, that is inactive enzyme form, and get cleaved and then becoming active enzyme. So C3 is becoming C3b and C3a, and then again the same pathway it's coming. So the final thing is the same for every complement system you can see, but the initiator is different. Okay. So we have talked about complement lectin pathway as well, where initiators are mannose and lectin interaction. This is one video of complement system you can see in YouTube. Regulation of complement. Complement components are capable of attacking host cells. Complement components undergo spontaneous inactivation if they are not stabilized with other components as its highly labile components. C3 convertase is a major amplification step in all three pathways. So this C3 convertase you have to know, you have to remember, because it's common in all three pathways, therefore that needs regulation. Soluble factors regulating complement. So yeah, this much you don't no need to recall all these things, but some important thing is that here protein S ligand or binding factor is C5b, 6 and 7 complex, and action is it inhibits MAC formation. MAC means membrane attacking complex formation, it inhibits protein S. And then again protectin, it inhibits MAC formation. So microbial pathogen invading complement system. So yeah, so there is system, there will be police system, but there will be some invasion by invaders. So there are different pathogens, and there are evasion molecules associated with them. They target some host factors and their mechanism of actions are there. It's very difficult to remember all these things, so just few example for you guys. Complement deficiencies. Okay, so deficiency of C1q, C1r, C1s, which is all are part of C1. C4 and C2 show enhanced immune complex and show SLE like symptom. Glomerulonephritis, vascularitis, these are different diseases where we can see deficiency of our complement system. Individual also show recurrent infection by pyogenic or pus-forming bacteria. Deficiency of factor D and properdin show Neisseria meningitis infection, but not with meningococcal bacteria. Okay. So deficiency of mannose binding lectins. Yes, tell me. Hello. Yes. It's disease. So SLE means autoimmune disease. Okay, where it deficiency of this complement system enhance some immune complex where immune cells detect our own cells and get killed. Okay, so SLE like symptom. SLE is a autoimmune disease. Yes, so you can Google and read about it more. Yeah. So Also, deficiency of mannose binding lectin show increase in respiratory tract infection. Deficiency of C3 show frequent severe bacterial infection. Protectin deficiency can be of high risk of thrombosis. Anti-C5 is used for the treatment of paroxysmal nocturnal hemoglobinuria. That's a big name, and come in the name of eclusizumab. Deficiency of C1 cause hereditary angioedema. Okay. So adaptive immunity introduction. So we will introduce adaptive immunity. So how different arms of immune system are linking. So there are cell components which can be non-immune cells or immune cells. They have pattern recognition receptors. Those will detect pathogen associated molecular patterns which are present in antigen. They will initiate inflammation, they will induce type 1 and type 3 interference and activate innate immune responses, and altogether initiate the pathogen specific adaptive immunity via B and T cell. Key cellular players of immunity. So the part of innate immune cells are neutrophils, eosinophils, basophils, NK cells, also dendritic cells. Adaptive immune cells are B cells, T cells, and there are some cells which acts very crucially in bridging innate and adaptive immunity. You can say those are innate immunity. Also, you can say they helps a lot as activating adaptive immune cells. Those are macrophages and dendritic cells. Comparison of innate and adaptive immunity. So innate immunity is already present in the body. Adaptive immunity you need to get developed throughout your survival. Innate immunity broad specificity, adaptive highly specific, fights with any living or non-living foreign entities, but adaptive develop highly specific immune response if not cleared by innate immunity. Innate is very rapid, adaptive immunity is slow. Innate is limited and lower potency, adaptive immunity is high potency. Time span every time the response is generated irrespective to the number of exposure. Adaptive, once immune response developed, the immunity can be lifelong or short, depends on pathogen. Innate immunity is generally inherited from parents and passed to offspring, but adaptive is not passed from parents to offspring, hence it cannot be inherited. Innate cannot react with equal potency upon repeated exposure to the same pathogen. Adaptive system can remember the specific pathogen which have encountered before. Innate present at birth, adaptive develops during a person's lifetime and can be short-lived. Innate none, adaptive immediate and delay hypersensitivity. Innate, there is no memory cells associated with innate, but adaptive immunity there is long-term memory. Innate immunity diversity is limited, adaptive immunity diversity is high. Innate immunity, alternative and lectin pathways, complement system activation, adaptive immunity, classical pathway. Innate immune cells present in skin, mucous membrane, temperature, pH. Adaptive immunity, lymph nodes, spleen. Composition is composed of physical and chemical barriers, phagocytic leukocytes. Adaptive is composed of B cells and T cells. Innate immunity development, evolutionary, older, and is found in both vertebrates and invertebrates. Adaptive immunity system has been developed recently and is found only in the vertebrates. Example, WBC is fighting bacteria causing redness and swelling when you have a cut. Chickenpox vaccination, so you won't get chickenpox disease because your immunity, there is generation of adaptive immunity and they are going to generate memory cells which will remember the foreign body in future infection. Yes, tell me. Divergence. Okay. Yes. So in innate immunity, there are specific cells which will act as classical pathways. But adaptive immunity, if I get infected with the same virus and you are also getting infected with same virus, there will be different kinds of antibody will get generated. So antibodies also, you have seen that there are different subtypes. So that means the diversity, the adaptive immunity is how you are going, like you are going to adapt it, and how I am going to get adapted. So this is diversity difference. So there are different antibody will get generated, so they are also switching, class switching will happen. So diversified antibodies, we'll discuss in next class that how class switching happens in different antibodies. This is called diversity. And limited means, yeah, limited means there are some limited cells will act in classical ways. So this is characteristic of B cell and T cell. B cell involves binary complex of membrane, Ig, immunoglobulin and antigen. Binding to soluble antigen, yes. Involvement of MSC molecule, no. B cell chemical nature of antigens can be protein, polysaccharide, lipid, epitope properties, accessible, hydrophilic, mobile peptides containing sequential and non-sequential amino acid. For T cell, we'll discuss this more. We'll discuss this factor in tomorrow's, like next day's class, in more detail. T cell involves ternary complex of T cell receptor, antigen and MSC molecule. Binding of soluble antigen, no, there is no binding with T cell. Requirement required to display processed antigen, mostly proteins but some lipids, and so on. What is the difference between immunogenicity and antigenicity? Immunogenicity induce immune response such as antibody production. Antigenicity not necessarily induce immune response but can induce immune response if appropriately modified. Hapten is too small, lack immunogenicity. These are hapten molecules. If you add some carrier molecule, the size is getting bigger. So if hapten is coupled with some carrier molecule, immune response can be induced against this haptens. This is called hapten carrier conjugate. Produces three types of antigenic determinants: antibodies to hapten, antibodies to carrier, antibodies to hapten carrier conjugate. So we will discuss about adaptive immunity antigen in our next coming class, next Tuesday. If you have any other question, we can discuss today. So Shilpa, you are in bachelor's, master's? Nice. Where are you doing your master? Okay, nice. So you are planning to come in research field or planning to do PhD? You will listen. So yeah, yes, do well in your master's dissertation. You will be doing in your um, parent college, or you can do outside in different institutes. Okay. Okay. Which question? Yeah. Tell me. Week 6 means is already due date. Okay, there is, due date is there. Yeah, tell me. Ye- yes. Both you can do. You can share screen as well. Yes. Yes, yes, tell me. Mm-hmm. Okay. Okay, okay. Okay. Yeah, yeah, yes, yes, yes. So agonist means which helps, and antagonist means which doesn't help. Okay. So we have to go through those information slides where I will see. It was there. The exact information I can remember, but if we, I can make you understand if we see the slide. The question what was the SLR? Okay, I'm just seeing. Hmm. So, if you, so you have found in the lecture slides about this, um, these terms. So yeah, so it's there in, yeah. So SLR14 is a RIG1 agonist, okay, which has been observed to induce robust anti-tumor responses in immunogenic or non-immunogenic tumors and can serve as a powerful immune adjuvant. Okay. So the question was, what was the question? Please repeat. SLR14 is a SLR14 is a, you can, okay, okay. Yeah, got got it. It will be agonist. Okay, it will be agonist of RIG1, but antagonist of what? Okay. Mm, hmm. If the question is correct, then we have to think that which is the antagonist of RIG1, then that will be the answer. And if there might be that there is just, they have written it antagonist but it will be agonist, there is chance of that also. Yeah. Yeah, tell me. uh rig 1 also is, yeah. Oh, so rig 1 MDA5 AM2 LGB2 TLR7 acts as a powerful immune adjuvant and induces anti-tumor response, is an agonist. I think it should be agonist. It should be this much straightforward. Hmm. Yeah. Yeah. Yes, yes, yes. Because they won't ask this much complex question. the it acts as like it helps rig one, but yeah, it should be agonist. You can mark rig one. Yeah. Yes, yes, it's okay. Yeah, it should be agonist only. Mm-hmm. It's okay. Thank you. Bye bye. Good evening.